Very good morning to everyone. Welcome to INBIOSIS online forum series number 5 for 2025. So I'm Dr. Azlan Muhammad, your moderator for today's online forum series. I'm very grateful that all of us have good health, time, and have the opportunity to attend today's session. For your information, this is the 5th online forum held this year by INBIOSIS. We have been collaborating together, organizing online forum series since 2023, and hopefully this forum and webinar series will continue in the future. We are honored today to host two distinguished panelists. The first one is Dr. Effirul Ramlan from University of Galway, Ireland. And second, Dr. Muhammad Akmal Remli, Director of Institute for Artificial Intelligence and Big Data, IBIC, University Malaysia, Kelantan. Both panelists are here to share with us their experience and knowledge in explaining and analyzing our interesting topic today. Will AI replace bioinformaticians? So before we proceed, I'll give a bit introduction of Dr. Effirul. So he is a lecturer at the School of Computer Science at University of Galway. He earned his PhD in Computer Science from the University of Southampton in 2009, specializing in computational and theoretical frameworks for nucleic acid computation. His research focuses on synthetic DNA or RNA interfaces and nanomachines for molecular level information processing in biological systems, with output spanning both computer science and the life sciences. Before moving to Ireland, he served as Director of Research at the MGI from 2017 to 2019, following a 10-year as Senior Lecturer in Artificial Intelligence at University of Malaya. He also previously had roles as a Genuity Science Assistant Professor at UCD School of Medical Science, Medicine and lecturer in Data Analytics at Ulster University. So he's been active in AI since 2003. He champions both applied AI in both research and teaching, bringing tools such as Copilot and Codex into his software engineering classrooms, and training students to use agentic automation-first workflows alongside sound engineering practice. His recent work bridges machine learning and biomolecular computing, from early nucleic acid information processes to current efforts on agentic AI applications in computational biology. So without further ado, I would like to open the forum to the session towards Dr. Effirul Ramlan. The floor is yours.
Thanks very much, Azlan. We have a bit of a connection right now. Sebab the island now, I have a PhD student that was Dr. Azlan former student, Chi. So it's very welcoming. And of course, I would like to thank Doc for the invitation, because anytime I can do an event dekat Malaysia ni, I'm quite happy, because it gives me an opportunity to cakap bahasa sedikit. So uh, it's quite different here, sebab sini orang cakap Irish kan. Alright, sharing my slides. Is it okay for everyone? Okay, I think okay, this is just a minor banter. It has nothing to do. It's not on a serious note, because I didn't notice it early on. But then when I was circulating the flyer around, somebody noticed that the image of my face was a bit doctored with AI. It was somebody who pointed that out to me. So I thought, if it's intentional, of a clever way of showing how AI could work, so that's kudos to the organizer. But at the same time, if it's in regards to how I look, so it's just a matter of beautifying how I look. So I can't do nothing about that. But if it's for the former reason, that's fits well with the session today then. Okay, I'm gonna uh look at what we're discussing in a different angle in a bit, which being represented by the titles that I have. Okay, that's actually a play on words on friends and enemy. I believe everyone knows that. So it's know your frenemy. Originally, it's from a song from one of my favorite bands. Okay, it's Know Your Enemy from Green Day. So if you don't know that, there might be a generational gap there. All right. And I'm always, as I mentioned, I'm always happy. Just to put a note that the calendar quite open now from December to semester two. So if there's any workshop or anything, especially the in-person one, so we don't have to be what 11,000 kilometer away. Do give me a shout out because my email is there on the slide then. All right. Okay. Straight on it then. So let's address the elephant in the room. I think the first few slides is about clarifying the misconception that people have with AI. I think the public perception of what AI is has been skewed towards more on the hype and the buzzword that goes with it. I mean, anything that goes into clickbait will be good to them, but actually doesn't really benefit anybody. The first one that I would like to address that seems to be a big deal is whenever somebody discuss AI, okay, that does not mean LLMs or just Gen AI or even a much narrowed down scope of GPT and even Chat GPT. So AI actually is an umbrella of research domain that covers things in a larger span. We started since 1950s, where guys like pioneer, like the father of AI, Alan Turing, or try to make computers that can mimic how human thinks. Okay, and if you look at where Gen AI is placed, okay, it's more we look at it more as a one of those killer app that has come up from this huge field of AI. I mean, there are a number of those that goes throughout from the beginning of 1950s until where we are now today. Okay, we have machine learning that can understand patterns in data early on in the 80s. And then there's a bit of a AI winter period, okay, where I was a part of that as well, where AI wasn't going nowhere. In fact, I decided to do PhD in a whole different field of molecular computing because I thought I'm one of the persons. So even when I was doing it, I did AI even in the early 1999s. Okay, even I was a bit skeptical of what's going on, and luckily, okay, we've survived winter, and by 2010 we got deep learning, and now the even more popular Gen AI as how you guys would see it. All right. So it sits, as I mentioned, as the umbrella of everything. So if you drill down, ChatGPT itself is one application representing what generative pre-trained transformer has managed to produce, okay, which is sits under the umbrella of what we have established, the attention layers that we see in large language model, okay, and so forth and so forth. So there's a rich history that goes into it. And it's more important to highlight that despite what you feel, okay, when you chat or when you prompt Chat GPT or when you prompt Claude or when you prompt Gemini and all the other fancy GPT based AI tool that you have nowadays, it's good to remember that it's good to put things into perspective. Actually, if we considered in synapses world, because that's how all the neurons are interconnected, okay, and we considered the perspective of how massive and how vast those neurons that we have in our brain, okay. So if we put that into perspective and we take even the, I would say, the largest, even though it's just an approximation, so the largest model that we have today, okay, and the ones that we can tout as the smartest, which is GPT-5. They did not close this information, so we did some approximation. So you have about like 1 trillion, 1 trillion parameters behind it. That is still only 1% of the total number of synapses that we approximate our brains to have. So if we put a radius, okay, this area of parameter, so that's only 0.1%. So they are, I mean, of course, it's remarkable what they can do, okay, but it's still lagging behind the total computational power that we can produce as a human being. All right. I mean, we might not be good at tasks that require fast calculation and things like that, but you have to remember when we started, okay, the first computer is just us, human being, relying on our computational power to do things. So looking at logical table, working out calculation itself, okay. So it is still human being that do all the reasoning and thinking. But yeah, I might need, we're not sure whether we'll get to that stage where we're going to have a GPT with the number of parameters to capture these intention layers up to the number of synapses that we have. But it might be to a point of singularity later on, but that's another point altogether. All right. Another misconception that I like to point out as well, because whenever we're prompting all this GPT, we get lost to the idea of that it can think and it can reason with us. But in actual fact, it is just still an algorithm that process token and then complete a sentence. That's it. It's clever in doing that. There's no doubt. It's very clever in doing that because it can work out a short range and long range association that have been calculated across billions of parameters and produce just the right word. Okay, so if I mean, despite what I showed just now in terms of the computational power that we have, so when we produce something, it might be like if you uh, ask me to complete a sentence what I eat just now and during dinner, so it might not be as fancy. Where GPT might then at the most extreme of the output will generate a total recipe of how to cook ayam rendang, for instance. Okay, I might just say I eat just rice and egg. That's it, logically, because that's what I remember. But then that's how far a snapshot of a memory that I have talking at 2 in the morning from Ireland right now. But a GPT might go to the extent and generate a total recipe. But that's because it's fine all the right association in generating the next token of words, the next token of word, and so forth. All right. Okay, so I have something of a kind of like a quick guide to how we get to the LLM that we see today. All right. So it's a demo that I built. Not that difficult at it because I always run Claude Codex with me. So this is a demo I built to show how we get from perceptron to then having multi-layer perceptron in the form of representative of deep networks, but not actually deep networks. Okay. Then how can we can build a simple sentence completer, okay, by just looking at a pairing of words. And then lastly, the self-attention layer that we see today. All right. So I'm just gonna switch my slide to this simple website. Wait a minute. Okay, so in the beginning, we have just perceptron. So let's say you vote for stuff, okay, and there's a boundary that gives you a decision of whether something is right or wrong. So defining this boundary of line give you the decision-making opportunity. So in statistic, this can be just a simple linear equation. So this is kind of like the model that you produce, all right? So if we train this, and if you have two input, which is the majority, your prediction output will be 1. So whenever you go below 1, it will then output 0 after it learns that model, which essentially just a linear equation, right? So then we move on forward. So we created a multiple layer perceptron, okay, which is just a simple unit that makes up the smallest part of a layer within a deep network, okay, because you have billions of those afterwards. So then we have the ability to train so that this network can learn something. So this is a simple multi-layer perceptron that will learn how a Boolean logic gate XOR work. Okay, we train it, so it produce this. And the key thing is this weightage, okay, that it captures. So that will be adjusted. So whenever you enter new input, it will then determine based on that pattern whether it's a 1 or a zeros. Okay, then you stack this up, okay, build multiple layers, okay, of this multi-layer perceptron and to a level where you have millions of those nowadays. And you can build something like a simple bigram model, okay? So in this instance, it will just capture the association of a pair of words. Okay, so if we check the top five of words that you see here, so if I put a context which is similar to how you would put a prop in, okay, it will then tell you how high its rank and find the most associated word with that particular context word that you put in. All right. So if you want to sample a sentence, it will then continue to complete things. Of course, it's gibberish because, as you can see, there it's only learned. Okay, we only build that by just using this simple corpus as a training text, not even extensive. So if you times this with the trillions of gigabyte data that you might have, then you can get something better. But in order to do that, it can't be a simple model. It can't be just a two pairing words to get things done, right? So it requires a more complicated architecture, and that's where attention comes in. So inside this small attention bit, you'll see how words are associated across everything. And if you look at this simple heat map here, it will show you, all right, how that word association will be important across the overall context. Okay, so then it will pick up that weightage and then become the words that you select, and you'll get something like a simple LLM. So I have a local large language a local GPT that I've uh, already downloaded, okay, under my Ulama server. So if you want to ask something like this simple sentence here in bioinformatics, a HMM is often used to call that model. Okay, it's connected to my local server running. Okay, it will then return back the output from that particular LLM of how we normally see today. So this is running on a lightweight model, but as you can see, it's already generating a bunch of stuff. Okay, all right. So that's a bit of a history. So it doesn't really look that smart because the once you understand what to build, it's there. Okay, so it's not really that complicated. All right. However, us in bioinformatics have taken leaps. Okay, it goes leaps and bounds nowadays. So we've there's a number of bleeding edge AI applications that has come up from this, and I think the two that's most cited and popular and kind of close to what I am doing is of course AlphaFold, now in the third generation, and another I find this is interesting because it's close to my research area. The one called RF diffusion from Baker's lab. Okay, that designs synthetic proteins. Okay, it actually leverages on the ability of this GPT model to hallucinate. Okay, and produce synthetic proteins. Okay, uh, so it's quite different in this instance because you guys can see we were discussing LLM and GPT quite early on, but actually, when we leverage the AI technology, so we are taking a lot more broader view. Yang kita dapat in this instance is actually not using GPT specifically, but actually using an AI technique that we call stable diffusion. So let's start with AlphaFold 3. So it's a diffusion-based model. Ideally, now in the third version, you can co-predict a full biomolecular complexes. It used to be only protein at the time. So when AlphaFold 1 and 2 comes up, it pretty much kills those people that works in crystallization. So it's no longer that difficult to solve protein. Still difficult, still value, but once AlphaFold 2 kill off all the competition in the biannual simulated protein competition, okay? It's pretty much a killer to those decided who want to do protein crystallization and their doctorate, okay? But of course, if that happens, it gives you more avenue to do things. So you might solve a protein structure faster, so you don't ended up doing too many X-ray diffraction and things like that. But it gets you to maybe to work on application, so rather than looking at structure, right? So then the way AlphaFold 3 work is quite straightforward. Okay, it learns by taking some the all the structural data that you have early on as part of learning, but for AlphaFold 3, it's been extended. So it will take all those molecules, okay, find the association between each molecule. And once you've break it down into tokens, all right? You run through what we call a simple term of gossip brains that will let every residue and atom talk to each other. So it gives a much larger interaction between them, okay? And then it runs through this designer, okay, that they call 3D sculptor that will implement the diffusion model, similar to how you run stable diffusion in DALI. Okay, it need to start with a set image, introduce noise, and then clean it off. Okay, so that's what's happening here. So once you have a clear picture of interaction of everything, then you introduce noise and denoise it to get to a possible complex. So that's what happened with AlphaFold 3. And as you can see from the tutorial just now, the importance of having this weightage association, okay, of the of pairing value across token, okay. So that's everywhere. So that's how data can be represented because at its core, these are still computer programs. They don't see structure the way we can see it, okay. We we can see things in a much higher level where you can easily identify patterns without going deep into it. But computers are still limited to numbers, okay, at its core. Even though you can done quite fast computation, it still see things as one and zero. So which is why all this embedding works, okay, in that sense. Okay, then another example is RF diffusion. Okay, it's a diffusion model that was actually built on Rosetta Fold early on. The Rosetta Fold work as a designer, so it allows you to specify what you want, and then it will then start off with a set image, hallucinate to what it will be. Of course, you specify constraint for it to work, and it will then clean that up, okay, from that noisy structure into a cleaner structure at the end. All right. So essentially, when you run it the first time, so if you can see in this panel, last panel here, it start with quite a noisy image, and then there's an evolution that goes, okay, but it doesn't work like evolutionary optimization or things like that. It works by just cleaning up all those Gaussian noise, okay, but this time done in the molecular phase, right? And you ended up with the structure that you want in the end. All right, so those are examples, but as you can see, one clearly missing bit there is you don't see any ALM or anything being applied. So this is something that I developed with in my lab. So we basically take an agentic AI approach because now those GPT agents at a stage where they are smart. But I'm putting an air quote whenever I say they are smart, okay? And they can pick up new skills and knowledge. So if we feed a bunch of knowledge. Like one of my research area is in fabricating a bunch of a new DNA nanostructure or nano machines. Okay, by using our own DNA polyamino shape. So it's a self-assembly mechanism that works quite differently because we use heterogeneous shape. And the aim is to allow my agent, okay, to take on knowledge of what I can do, and then if given a structure or seed structure that we inspired to be, it will then map out how those blocks will be placed. All right. And then from there, hallucinate to come up with something close to what you want, but a bits and pieces that need to be changed. Okay, depending on the functionality of such structure. All right. So we haven't yet completed the stable diffusion part to allow our GPT to hallucinate. However, the mix and match of placing this block can be done rather straightforward in a line of code that was a lot less than how you would do it via dynamic programming and things like that. So that's what's happening in that space. So it will feed all those knowledge in and refer to each knowledge and what's there, and we can limit our GPT to only function within that space. So that's the context that we restrict it to be. And then it helps us to do this mix and match that will then fit as an input to a stable diffusion system that will clear and clean things up. All right. So if without, I would say without, I mean, without an agent to do this, okay? If it's require a grad student to do it, I think I might need two, three students in the grant. So over here in Europe, that might already require a funding of, I mean, across 4 years PhD program. That could be a year, would be 1 million. So which is quite difficult. But by having just credit to Open API, we have this pipeline set up, I think, with less than 30 euro. So in terms of cost and what they can deliver, because you can just run this all the time and they can work 24/7. So it's quite a good way of getting a complex task to be completed, okay, by a machine rather than by AI, in fact, instead of a grad student, right? Okay, so what are the drivers, okay, that allow this to happen? Because I actually had this discussion with a bunch of my colleagues in biology, because we are aware that PDV was set up 30 years ago. I mean, there's about already 100,000 to 200,000 structure by now. But how come we didn't get AlphaFold early on, right? Well, what's stopping us? And the one thing that made it possible, so despite the rich and organized structure that we already have, we couldn't get to AlphaFold because the algorithm was not ready. So this is one of those rare instances where computer science actually need to catch up. Okay, data is there, all the structures are well documented. You have NMR base, you have X-ray diffraction, you have cryo EM type of structure, but people have tried to build this predictor, but it wouldn't work because you need something more, something that can understand pattern at a whole other level. So you're defining nonlinear relationship in multiple direction. And I think because I have the godfather of AI there, uh, you have Hilton, Lenkun, and Baggio there because they keep on working. Okay, with the generous contribution from the Canada government, they keep on working. They didn't stop. So during the AI winter, where we feel neural network was doomed, it wasn't going anywhere. The three of them decided to soldier on. Okay, so one of the I think one of the uh, game changer was when AlexNets, okay, which is a form of deep learning developed by Hinton and his PhD student, outperform all the other image recognition model, okay, in the image net, which is an Olympic for computer vision, all right. And then Yan Lekun keep on working and establishing a better CNN module, okay. And then Benjo continue understanding the layer of that you required in order to make a proper deep learning networks and all. And they have been there's enough recognition. I think everybody in the community of AI know these three people. Okay, the Godfather have been awarded the Turing Award, which is in the field of computer science is the Nobel Prize, right? So that's but that wouldn't be enough though. So even though now we have the complexity that grows into uh that was equipped with attention layers, it becomes transformers, then it becomes LLMs as how you see it today. We still do not have the computing power to process all this. So another drivers that comes in, okay, that comes from unlikely places was the GPU cards with CUDA that allows you to run things in parallel. I mean, we need this because one of the benefit of our brain is we can think in parallel. Okay, everything runs in a parallel mode, but it's not how we normally design computers. It's always works sequentially. And luckily, through gaming, because they require all the processing power to render things, okay, higher refresh rate and things like that. So they have a parallel processor for gaming, which is all those GPU cards that you have. And then NVIDIA decided to build CUDA, a layer to code things in parallel. So that allows for all this weightage calculation and vectors to worked out, because vector can be seen as the smallest unit whenever you do something in deep learning, and there's no way you can do that in I mean, you can, but you gotta wait years then. Okay, so GPU allows that to happen, and that grows. So even now, you can see data centers being built just to run or fine tune this model, right? But the thing that I like about NVIDIA, that they they put a proper name for each of their data center. They call them Stargates. Okay, so that's, I mean, if you know Stargate, it's kind of like a portal that open you to different dimensions. So that's what they are building. And if I'm not mistaken, the biggest Stargate data plant is in UAE, in fact, that have the capacity to do 5 gigawatts, which is a lot. And lately they just recently they made an announcement, a co-funding between the major player, of course, NVIDIA and OpenAI, to build a 10 gigawatt data center. So this is not good in terms of sustainability, because a 10 gigawatt data center actually can power one third of the whole of Ireland. That's around, can't remember the actual number, maybe 100 million houses in the US. So, so the it's not really green then, this technology, right? And then another drivers that allows the boom in AI to happen is the software 2.0 mindset. So we accustomed to the way how we build software, where you need your data, you need your all your codes, and then all the processes to happen. But then the mentality of software 2.0 is you just need to set up data and you just need to set up your target. So once you select a model, that model will perform all the processing for you. So the output of such model will be all those vector and matrices of weighted that has been adjusted for you. So all that parameters adjustment will be the coding itself. So the way we change our mindset in terms of how we want to develop software allow a much prosper and encourage a lot more development in this space, which facilitate what we are seeing right now in terms of bleeding edge AI that we can produce. And lastly, the openness of the community. Because I think at some point we realized that in the space of bioinformatics, there's a lot of things to pick up. All right, it might be it's always the conundrum of whether you're taking a biology student and teach them computer science stuff so they can program, and then they can learn statistics in order for them to do something, or then you can take a computer science student or teach them all the biology. But with the ability of AI right now, you can create all this pipeline instantaneously. So it eliminates the need to have an actual student to do all these things. Okay. In fact, you yourself can function as a supervisor to do it. All right. You just need to supervise things, make sure things run properly, and the willingness of the field to embrace and accept AI is it is one of those drivers as well that allows AI to prosper pretty much in the field. Okay, do I still have anything left? All right, so I'm gonna close out with a few things. All right, just the words of wisdom from fiction and non-fiction people in the community. One is from one of my favorite physicists, Richard Feynman, who say, "What I cannot build, I do not understand." Actually, it's a flip of that in terms of what we discussed today, because I think a lot of the misconception that occurs because we do not understand how the technology work. All right, and some people are saying that we are in AI bubble because they are seeing a number of industry that first going gung ho when they when they adopt all this AI technology, decided to then go back to their normal or typical ways of doing things. But basically because they were fascinated by what AI can do without understanding actually the whole inner workings behind it. So if you understand, I mean, it doesn't have to be very detailed understanding or comprehensive understanding. You don't need a PhD to understand really what's happening behind it, because it's it's already there on surface. So if you understand what it can and cannot do, so it gives you a ground of what you want to develop, and you understand the output, and you know what to do to make sure you get things right and things like that. All right. So it's important that, and because we can build all this, so it's not rocket science or anything. Okay. And most of the time, if you understand what you're doing, you should have a good results, right? Then another one from a fictional character, all right, Master Uguay, is to embrace this. Okay, yesterday is history, tomorrow is a mastery, but today is a gift. So rather than, uh, I would say, instead of being stuck in the old ways, because I'm not into early on with all the buzz that's going on behind GPT and things like that. I'm a bit skeptic as well because I've experienced it, try run a few things, and it might create beautiful output from the offset, but actually if you dig deeper you find a lot of flaws in it. All right, similar to how you run things via DALI, then you realize there's an extra hands or extra fingers in your hand and things like that. But on the initial outlook it looks great, but where we are today, I wouldn't say it's mature, but it's at the state where you can do something with it. But I'm from the camp that believe if you design a specific purpose application, then it works a lot better. So if you restrict things and it works only on the context and domain that you define, I believe you should get the result that you want. So you'll be able to build a system that works in whatever things that you want to accomplish. All right. Okay, and lastly, because we discussed frenemy as the initial topic, right? So kawan ketawa senang nak cari, kawan menangis susah nak jumpa. So I believe my relationship with AI is still only on the fun bit, right? Because do remember, so despite with the current version of GPT, you keep on getting those encouragement work, like because OpenAI try to make GPT in that instant, or even Anthropic Cloud, to have a feeling and understand what's going on. But as they don't have a brain, they don't have a heart as well. So they don't really understand what actually you are feeling and things like that. So if you know how to build things properly, right, you definitely will enjoy it. And even though it's still a love-hate relationship, even to me, because at some instance, you feel like you just want to get something going, and once you give it enough context, you want things just to be developed for you in an instant and it works all the time, but that's not always the case. Right? So uh I think that's all I have. Thanks very much for your attention. Return the floor back to Doc then.
Right. Thank you, Dr. Effirul. A very good introduction to the field of AI. And basically, you sum it up right. What's the current progress, like the intersection in between AI and bioinformatics, and you give a glimpse lah on what's the way forward, the certain gaps that we are still having at the moment. So for those who have questions, just list them in the group chat. We'll have the Q&A session after Dr. Akmal's presentation. So let me give a bit introduction for Dr. Akmal. He's the director of the Institute for Artificial Intelligence and Big Data, or IBIC, at Universiti Malaysia Kelantan. The institute, recognized as a center of excellence, focuses on advancing research, development, and training in the fields of artificial intelligence and big data. So his primary research interests include artificial intelligence, data science, business intelligence, and computational systems biology. Under his leadership, IBIT has secured over R1 million in research grants, focusing on AI applications in bioinformatics and metabolic engineering. Beyond his academic work, he also have many collaborators with industry partners and engages in local and international research collaborations. He's also currently a visiting research fellow at the Center for Research, Innovation, Development and Entrepreneurship, CRITE LT, in the United Kingdom, and as an advisory member of NOS, a catalyst for European cloud services in the era of data spaces, high performance, edge computing under the Air Institute Spain. So without further ado, I pass the floor to Dr. Akmal.
Okay, Assalamualaikum. Very good morning. First of all, I would like to thank you to Dr. Azlan and the organizer for inviting me to share our knowledge or our journey in AI that related to bioinformatics. Let me share the slide first. Okay. Can you see the slide, I think?
Yes, Doctor. Very clear.
Right. Okay. First of all, actually, I just recovered from influenza A. It's very, very, the feel is very. I still can feel the effect of that. Maybe disease. I'm not sure the bioinformatic terms. Yeah, but my journey actually in UKM, Dr. Azlan, in UKM, I have two institute that very close to me actually, close to my heart actually. One from UMBI, Institute of Biomolecular Biology, that focus on the disease kind of cancer and so on, and also INBIOSIS that focus on the systems biology parts, computational system biology. I think those two institutes are very close to me because it's maybe related some of my research area. Okay, actually, I'm not disclaimer. I'm not the expert one of the bioinformatics. I share some of the field with Dr. Effirul. We are in the computer science people. But I started to do some research in my master degree in 2009, doing the gene ontology. Gene ontology combined with the metabolic pathway. And at that time is my master to set up the knowledge-based system. At that time, no LLM, no ChatGPT, and so on. We set up the knowledge-based repository. For example, to answer the which gene are participated in some specific pathway. But we are computer science. We are developing the framework, the methods, the algorithm, and so on. That one during my master. And then during my PhD, I do a lot. I do in the field of the computational system biology, but we are developing the optimization methods to find or to locate the kinetic parameters in the what we call I just forgot, in the bioreactor or in the micro metabolites, the we want to fit the parameter into the experimental data, so we need to have the optimization methods. So starting from that, I still continue my research in that field because that's my main area actually. So I moved to UC Malaysia Kelantan. I didn't have the partner. That's the very challenging actually. So because the UMK is different from UKM, so at the same time I need to look the other research area, which is now I also embark in the business intelligence, or we want to build the computational model to do in the financial market. But I keep both of my research because the biology part is also my research. Until now, alhamdulillah, we are still, even not too much, I still attach some of my research in this area. Okay, that's a little bit about me. So now I'm serve at the Institute for Artificial Intelligence and Big Data, IBIC. So my faculty is a faculty of data science and computing. So IBIC is the COE that registered under the KPT lah. Okay, actually, I already quite impressive with the Dr. Effirul presentation because that's actually very advanced. You know, Dr. Effirul tried to present the computer science, which is very, very, some of knowledge is very advanced in the very, very, interesting way to how to get used at, or how to know a little bit about the LLM, generative AI, and so on. But in the bioinformatics, we all know actually, bioinformatics for me as a computer scientist, it's a very challenging field actually for me when we start the research, because as a computer science that didn't have the background of the biology, we have to understand maybe not too deep, but just to understand the overall picture, how or why we need to do the research in this area. Okay, actually in bioinformatics, for those who are not familiar with, we already know that it combined different or multidisciplinary area like biology, data science, and algorithm part in the computer science part, computation, and so on. Okay, until now, in term of we see that a lot of the technology advancement, especially in term of the LLM, large language model. We can see that at the certain now the certain area, the bioinformatics AI only can recognize the pattern. We have the million of data, heterogeneous data that a lot of noise and so on, and AI can excel to uncover or to detect or to find the pattern, to find the hidden pattern and so on. But overall, it cannot understand the biology. Same with the LLM. If we know the detail of the large language model, maybe using transformer deep learning, they all just statistical method. Of course, they excel or they have very, very smart to detect or to produce the words, but it's all just statistical model. So it have the limitation to understand the overall biology parts. Okay. For example, AlphaFold can predict a 3D protein structure, but doesn't know the protein's function, the stability, or disease contact, because it needs still the human to in the loop to validate, to to do some of the research and so on. Okay, so we try to see for example, that the Nobel Prize in the chemistry 2024, we have in the field of protein design and protein structure prediction. Okay, for example, David Baker in his field is the computational protein design. So he and his team are involved to propose or to design the new proteins from zero using then AI. We call it the model of Rosetta model, and also deep learning tool protein MPNN and AI-based method, deep learning method, which is called family-wide hallucination. And other two guy, Demis Hassabis and John M. Jumper, also contribute in AlphaFold 2, and also that project to predict the 3D molecular structure from 1D sequences using artificial neural network. Okay, artificial neural network is also the AI methods, which is in the umbrella of the deep learning, and also using transformer-based model, specifically known as Evoformer. So it's use the latest breakthrough from the AI parts to design the new or to propose the new breakthrough in specific context. For this example, in the protein field. Okay, Evoformer require about 80x million of parameter. Now we are dealing with a very, very large model with a million of parameters need to be tuned. Okay. AI actually change bioinformatician works, not why they are needed. So those who are in the field of bioinformatics or just want to enter in the field of bioinformatics, so you can use AI maybe to accelerate your research to find new findings in terms of the biological phenomena and so on. So you must use the advance of AI tools nowadays that you have, so to design in your research to produce more output for your research and so on. Okay, maybe we want to know the personal AI. Maybe in if you are new to AI and want to dive deeper into the bioinformatics, maybe this one will help you. Okay, I think a lot of cases or business, what we call the research is in bioinformatics is very large. Actually, it's, the field is very large. We have a lot of like the detect protein to find the apa, drug discovery, to find the biomolecule and so on. Okay, for example, structure prediction, you can use AlphaFold 2, ESM4, and for the code automation, of course, we can use also the Gemini, ChatGPT, Blackboard, BioCopilot. And drug design, we can use Gemberta, Moleformer. Data analysis, we can use a lot of the tools nowadays, Google Colab, VS Studio Codes, Jupyter Copilot, and so on. And literature, we can use Scopus AI and SciSpace. Okay, this one is for your reference. Maybe you need to go deeper. You no need to tackle very large field of bioinformatics. You need to tackle just small amount of the small area of the research and try to excel in that area. Okay, I think Dr. Effirul already touched about the cloud and about the infrastructure that actually we needed in the research. But I think we are not competing with giant company like Microsoft, NVIDIA, and so on. We cannot compete with them. That's for sure. We cannot build the new one that especially that compete with the we cannot do that. Besides, we must know our needed. Do we need a local computer, which is the spec is somehow can accelerate our research or can speed up our computational part, or do we need the cloud services? It's actually depend on our problem and our background. Okay. So the example for the cloud, so you are running AI models in the server. For example, you can subscribe the Google TPU, Amazon Web Services, Microsoft. So you can rent that server. They have many package that you can rent. So you maybe you code into your local computer and you deploy or you compute the task. For example, to train the model using the cloud services. Okay, that's the cloud-based service. Also, if your lab have some computational power like GPU and so on, so you can try to build your AI model using your own local computer. The workstation HP. I think most of the research have that infrastructure, but not too not too very good or not too very high spec in terms of. But if you can do that, you can utilize that. Please utilize. I give the example during my PhD in 2017. I do a lot of the estimating the parameter, about 100 of parameter, but the computational parts require a lot of time. One CPU at that time needs about 3 days to compute, to optimize my parameters. So I need to speed up actually. So at that time I asked my supervisor, I want to use all the 6 computer in the lab. But fortunately at that time there no one used that computer. So I set up the parallel computation. So we can simultaneously or parallelize our computational method. So from the overall, if we run the model in one computer, it can consume 1 week. Okay, by using the 6 CPU or 6 computer, I can reduce my computational time into only just 2 hours completed. So actually that's a very significant for me. So I can speed up my research even though I'm still in the Langkawi at that time to holiday, but my computer is still running, compute to get the result and so on. So when I come back from holiday, so everything is finished, and I just take the value or the numbers to be put into the paper and to the thesis. Okay, that can speed up our research, but it's depend on the context first. Okay. There is advantages and disadvantages of using cloud AI and local AI. For the cloud, you no need to buy or to maintain expensive hardware, but you also can increase the computing power instantly based on the package provided by the provider. Okay, you also it's easier for teamwork and collaboration if you're using the cloud AI. But there are some issue lah. Kan kita sometimes we must pay attention to our data. Maybe our data is not, no one have our data, so you must protect your data. Data is very important. Like sometimes you collect the data, raw data in biological sample and so on, but you put into the like the ChatGPT or to other AI-based tool, actually it's exposed to leaking of your data. So please be careful. For example, especially your data is very, very new or very raw. Okay. In terms of the cloud disadvantage, also it depends on the cost because it's not cheap actually to subscribe or to rent the horsepower in the cloud. And also it's required a stable connection lah. For the local, we have the local at IBIC we have the local CPU or local HPC, but it's not too good. It can somehow better than laptop. Just to to test the model, to deploy, and maybe to left the model train using the computer at the lab, maybe in the weekend. So after we come back to work, so already finished. So that's the advantage if we use the local computer. Data privacy will be secure more, and then no internet required. Of course, there's tak perlu internet lah kan. So but the disadvantage, we need the powerful computer because it's harder to scale or to deal with a very, very large data set because the biological data is very large and huge. Okay, and also it's need to maintain and software or hardware setup. For example, if we go for the cloud, now we are there are a lot of tools nowadays to analyze the data in the cloud. For example, Google Colab, but you need to do the coding. Amazon, Hugging Face, and so on. For the build our own local AI, we can use the MATLAB, Jupyter, BioPython, and so on. My research in that time, I'm using the MATLAB only. Okay. So in the Malaysia context, okay, the bioinformatics actually, yes, they are limited in the infra actually, computing resources, and at the same time, the shortage of interdisciplinary talent. But is this thing actually because we have AI comes in that can be, can be speed up the learning process actually. So maybe Dr. Effirul already told that. For those who are not in the computer science, actually you are entering the new era of AI that you can skip some bit to learn about the computing parts. There's a lot of AI agentic AI, then maybe can speed up our research. Okay, but actually we still need the expert for those discipline, the computer science part and also the biological parts. There's a very rare is not very tak ramai lah kan, tak ramai sekarang ni di Malaysia. And then you need also the high cost of cloud computation. Okay, so that's the the challenge now that in Malaysia, that how we want to accelerate the scientific discovery by using the bioinformatics tools. Okay, for those who want to excel more, you can use a lot of open source tools like the BioGPT, SM4, DeepChamp, and so on. So I encourage also everyone to join cloud-based academic collaboration. We have a lot of the resources, for example, in the GitHub, Kaggle, and so on. So publish your work also. Then you can collaborate with other researchers in the overseas and so on. And also, you need to the computer science. We are the computer science people. We are heavily depend actually from the public repository, the NCBI, EMBL, and our new release of the Polar Bytes platform. I will show you after this. So public repository is curated, so you can use those data to do your research. And also, if you have or other institutions have the CPU or high HPC, so please try to collaborate. We don't want to do the research in silo. Okay, try to collaborate, use their platform, and then uh, in the same time, you can do the research team, research work together to do the their publication and so on. So this is very important to overcome the limitation in the bioinformatics research, especially when we are using the artificial intelligence. Okay, I just want to share some part of the. Actually, this is the first time I share about one topic that all of you must know actually about the degenerative AI. So all of us already know the word of generative AI, but I want to share some little bit about degenerative AI. So that's the opposite of generative AI. So everyone know that, and why this is matter in bioinformatics. Okay, so everyone know that generative AI can produce the new data actually, the new uh, the new data involve of the attached image. In terms of biological, we can say that can generate the protein, molecular structure, and so on. But in the basis one, can create the audio, the image, the video, and so on, the text, and so on. That's the generative AI, like the ChatGPT, the Gemini, and so on. Okay. The opposite of generative AI is a degenerative AI. So what is that? Actually, a degenerative AI, or also we can call it a model collapse. Model collapse is occur when AI systems are repeatedly trained on data generated by others AI rather than real human created data. Actually, the generative AI, Gen I, create the new data, okay? But somehow later, maybe the new AI system will learn the data from AI. So AI generate data, and then other system of AI will train their system using the data generated by AI. So that is the the originality of the data will be decreased because the data is synthetic. We also use AI to learn about the data generated by AI. So that's we call degenerative AI or model collapse. So why is matter in terms of the bioinformatics? Okay, if you look at this graph, okay, this is actually the contents, to be specific, the articles written by AI has been, has been surpassed by the human. Okay, AI generated article now exceed human written once on the internet. So maybe we don't know. 20 years later, we didn't see the original content or the article written by the human anymore. We don't know. Okay, we don't know. The internet will flood with all the content generated by AI. So the consequences is it will somehow happened to the AI, which a model collapse. The nature is the first generation AI learn from real data, scientific papers, biological database, protein sequence, and so on. And it will produce, of course, it will produce useful and realistic output. That's the first generation. I still remember in 2021, I wrote the grant, FIGS grant, using the generative adversarial network, try to propose the method to generate synthetic data. We use gene expression data combined with other data at that time, but unfortunately, that not my application not get funded at that time because 2021 just we are starting in the era of generative AI at that time. But I tried to do that, but not funded. That's the way we generate the new synthetic cell. Okay, because the limitation of the data is hinder us from publish or to find a new discovery in our field. So we need to have the synthetic data. How to do that? We can use the generative AI. Okay, on that time, if we have a lot of generate data from the AI parts, it will go to the second generation, which is the next AI model is trained using the output of the first model instead of original data. It will lead to error, biases, or simplification from the first model. They are now back into the training data. So we generate the data, and when we use the data to train to the AI models into the second generation. And after that, after several generations, the model start to forget what real data looks like. Okay, this is actually, we must have know about this phenomena actually. Okay, what might happen if we generate a lot of data and then we use the data to generate the AI. Okay, this is we call it model collapse. Okay, this is my sharing about this lah. Okay, this is for example, you can see the image on the right top on the right screen. There is the sample that we generate the picture of the man. After that, we generate again, and then in the last day, we didn't know what that image exactly looks like. Dia dah jadi collapse. That's the model collapse. Okay. So model collapse is making a photocopy of a photocopy. So we for those who are in the field of bioinformatics must know that this might be happen in the future if we didn't care about. Okay. Yeah, because it's a matter actually in bioinformatics because our data authenticity is critical. We rely on heavily depend or data experimental, which reflect real biological complexity. In future model, use AI generated synthetic sequences as training data without verification. Error will be compound and distort scientific conclusion. Actually, okay. And then if we use the degenerative AI, it produces a structure that look plausible but lack of biological stability. Okay, it will cause to reduce the ability to discover truly novel biomolecules, and then impact on biomedical discovery. It will limited our finding if we use a lot of the synthetic data. In the first round, if our first generation is okay, but after that we must be careful where we use our data. Is it the synthetic data or the real one? So that's some sharing of my degenerative AI. So no problem. It's okay because you can use the data from the trusted website, for example, NCBL, EMBL, PDB, and Polar Bytes. So also you can also do the collaboration works. So you try to collaborate with maybe the computer science people that working a lot with the data. Okay, the integration of data, the authentication of the data. Is it the data generated from AI or not? Okay, kalau kita tengok nowadays dan lot of video are generated by Sora 2 or OpenAI punya algorithm. It's very realistic. So somehow we cannot detect this is the real or not. That's actually the dangerous of the data replication or data generated by AI. So same with the scientific field. Okay, I want to skip this one. Okay, this is I want some share some of the related publication that we just published in IBIC UMK. Maybe it will give the benefit to you. Okay, this is the we do the review on how the generative AI Gen I can transform drug discovery. Okay, you can go to this QR to read more about the detail of this paper. This we collaborate with the Dr. Chin Siok Fong, also one of the collaborator of this research. Okay, this one is more to the proposing the dense net, one of the algorithm in deep learning that used in the CNN, convolution neural networks, to predict lysine crotonylation, okay, site in protein sequences. Okay, this one is we produce also the methodological paper. We develop polar bytes. This one is research under the Yayasan Penyelidikan Antartika Sultan Mizan. Collaboration also with INBIOSIS with Dr. Sarahani. We publish, we develop the polar bytes that where the data that we collect from all our resources contain the polar region data we integrate, we put into one umbrella to make sure the researcher can access or to do the further research. Okay, this one is published in environmental modeling and software. Okay, I think that's all my sharing from me. Over to you, Dr. Azlan. Thank you.
Right. Thank you, Dr. Akmal. So basically, we already have a few questions. So I also invite Dr. Effirul as well. I think the first one is I think Dr. Effirul might be able to answer that. So so currently, IFA has a workflow for atomer-based biosensors, and they are using a pool of DNA sequences derived from tRNA databases for AlphaFold3. So they are inputting them into AlphaFold3. But there seems to be an issue of random and lack of clear structural convergence. So what's your advice in order to improve that?
Okay. All right. Actually, this is very close to what we did. We've stumbled across the same problem as well. So once Alpha 4 release and then Alpha 4 3 release, we kind of figure out that this could be a good way to simulate things. But then once we understand that actually when Alpha 4 3 make a claim that they can do a full-on DNA or RNA or protein RNA or protein DNA complexes in the as a generated output, we figure we kind of like we play around with a bunch of DNA nanostructures to see what comes up, and apparently we have some issues with what's being generated, especially in the nucleic acid space, and that comes down to AlphaFold 3 having limited data when they train it. Okay, so I think that's what you will encounter as well, because there's a limited knowledge when they play around with complexes that's not entirely protein. So dalam group saya benda sama jadi. So what we decided to do is we actually build another layer of architecture on top of that, and we pretty much get a pipeline running where you do all the basic binding affinity calculation and base pairing and things like that by just using Oxford DNA or XDNA. That's one layer. And then we create another layer where we fit in our own data set of nanostructures, all DNA and RNA based. So we created that data set. So we have around 300 structures, and then we overlap it with nanobase.org data structure. So then it but then at the same time we created an agentic AI that work on that space as well. So so given this whatever that you wanted to design structurally, so we also encode it in a language, structural language that we understand. So it will fetch in and out against the database that we have, and then pass it back to AlphaFold 3, and then they do some, there's a cross reference that's happening there as well. So I think to solve that problem, ideally, you can't rely entirely on AlphaFold 3. So AlphaFold 3 are good if the input type are all motifs based. So if it's a more higher level definition, so it's more like a structural motifs within tRNA binding sites that you want, okay? Binder pipe and that region and everything. So they might be able to give you a good approximation. Tapi when we discuss just lumping the whole sequence structure database to AlphaFold 3, you might not get the expected result. And it's quite common for them to veer off path and produce those things. Tapi we realize that when you look at it closely, it's easily fixable. So for instance, we've thrown in like a structure of a, I can't remember what was it. Uh, it was a prism of some sort, tapi it's a symmetry. So they are the motifs that we want, okay, uh, were identified by AlphaFold 3. However, I mean, the covalent bonds and how those stretches of duplexes that should be a major thing, they kind of skip it. So, because there's no, okay, one thing to understand as well, AlphaFold, especially if you're using DNA or RNA, there's no energy model behind it. So this approximation that's being generated by the model are just understanding of how bases should sit to index next to each other. So let's say if you have a clumps of A, G's and T's, and this is what a motif would look like and how they should bind. Tapi there's no energy model behind it. So we realize that you still need physics to ground what should pair and stuff. But then if you give it a much higher level information as input, then normally approximation will work as you expected. So that's what we did in terms of working with a non-protein molecule on with AlphaFold. Okay, but I think if you, I probably need a bit more detail to understand. I mean, maybe what the target binding site that you're looking at and things like that. So maybe we can discuss further if you're interested. Okay.
All right, thank you, Dr. Effirul. So let's go to the second question. From Dr. Murni. I think let's have Dr. Akmal answer this. So how ready are Malaysian institutions and industries to integrate AI into bioinformatics research, and how we can ensure that AI generated bioinformatics results are trustworthy and reproducible? So maybe some thoughts from you.
Yeah, how ready Malaysian Institute integrate AI into bioinformatics? Actually, bioinformatics itself is actually we cannot separate with AI. A lot of parts of bioinformatics actually from AI. From in the early we do the protein docking, and then for example, genocard. There's the some part that in my journey that we naturally use the machine learning. For example, during my research in my PhD, I attached in the Spain's lab doing the cancer classification. We use the data from colorectal cancer. Okay, that's the naturally we use the AI algorithm like machine learning and so on. Actually, the bioinformatics already embed the AI in the field, but now with the advancement of a lot of the algorithms LLM, we must progress to the new embark the new technology or the new things. So that's the we must learn progressively learn, not doing just the old one. We must doing the very, the advanced field. If we are looking, we are computer science. If we want to do research in area in AI on the biological data, we need to refer to the Web of Science paper. So at that during that process, we know that what is the currently in the other outside Malaysia doing. So we can somehow know the advance of the AI application, what is the state now they are doing. For example, in 2021, they are just new to produce the synthetic cell using the generative AI, generative adversarial network, and so on. But now a lot of things happen, so we must keep up the the current research now. So we need to read a lot also what is happening in the current stage of the AI. Okay, that's for the readiness of the AI in bioinformatics research. In terms of the want to ensure that AI generated bioinformatics results are trustworthy and reproducible. Okay, actually this practice is very well did by the UK and overseas research actually, but in Malaysia context, we are still lag behind. For example, if we do the experiment research using the biological data, we must try to to well document the process. The data must be well documented, so we can also have some kind of the platform to reproduce our result or to reproduce our experiment, so others researcher can take that benefit. So we lacking some of that practice actually to make sure the result are good or can be reproduced. Okay, so it's depend on the researcher. So try at least for those who are doing the AI, much of the work in AI in bioinformatics, try to do the quality research. Your research must be in a high quality and using the standardized data, well documented process step by step, put into the lot of the public repository like the GitHub and so on, then produce for others in Malaysia or other collaborator try to use that or try to reproduce that result to promote, so we can make sure the process or the result is trustworthy and can be reproducible. Maybe we can start that, the good practice to do that. Okay, that's I hope that I can answer the question, Dr. Moni.
All right. Thank you. So actually we have less than 10 minutes, so maybe we try to be quick going through these questions. So the next one is from Vino. So I think I pass this one to Dr. Effirul. Basically, how can AI be integrated into a compound database in order to do prediction of compound versus targets? So what's your thoughts or just some ideas on where to start?
Because I like the word network pharmacology that you
Yeah.
have there. I mean that's a good thing, and straight. I mean, it directly, you probably can come up with a graph neural network, a GNN method, where you map out your plant compound, what its target, the affinity, pathway disease method and stuff to build an overarching network of everything. So it is quite a natural fit whenever you do network pharmacology. So it's easier for you to just weighted stuff, so you can easily do prediction and stuff. Another way, if you want to be fancy a bit, tapi it might be a bit more computational work, but you can be done. It can be done. You can build a deep drug target network or deep drug affinity network as well, in order to help you to work out the chemistry and how the sequence relate to chemistry, and then get the compound target as well. So that's another fancy way to do things. And the most basics could be run a multimodal machine learning that using your omics data, quite straightforward to run. So it's no longer complicated these days. I mean, you can get a multi-omic ML pipeline ready in 10 minutes, I believe. Okay.
Right. Thank you, Dr. Effirul. So I think we'll combine the last three questions. So mainly about hallucination and the second one is are we actually going into the generative AI? So from Dr. Go, Dr. Chin, as well as Dr. Bello. So your thoughts, Dr. Akmal?
That's actually the masa depan, the future of AI. Actually, we are entering the phase of uncertainty. Actually, to be honest, from my perspective, actually we don't know where are we going in the maybe 20 years later or 30 years later. This happened because there's the sudden change in the AI direction since the release of the transformer, the researcher from Google DeepMind that produced the attention is all unique paper. From since that one they built the GPT, ChatGPT and so on, and it directs into the some extent of the AI to generate a lot of text data. Once we can generate a lot of text data, we can generate a lot of things, a lot of the application in very, very different domain, for example, in biology, in agriculture, and so on. But yes, nowadays we are talking about the agentic AI. Okay, agentic AI to to help the organize to solve the problem and so on. So that's the we are heading at. But in terms of degenerative AI, is some kind of the still in the the things that we must aware of. When all of the data generated by the AI, it might be happen. Okay? So that's the I personally cannot predict lah apa yang akan what will happen in the next couple of years, maybe 10 years or 10 or 20 years. But we are entering the era of uncertainty. We don't know, maybe 5 years later there are some the great things will happen in the AI. We don't know yet, but that's the currently we are heading. So we must make sure the most important things about the AI is the data. So if we have the data, we must protect our data. That's for sure. So yeah, hope this can answer your question. Sorry.
All right. Thank you. So basically, I try to sum up our sections, right? So from the two speakers, basically, they gave us an overview, how it comes to the current state of today. And actually from both presentations, like I can see that, actually, to me, we need actually more bioinformaticians. So AI won't replace us, because we now see the gaps and the main change is basically how we can evolve, right, to utilize all these tools and everything in order to advance biology research or systems biology research in the future, right? So take advantage of the capabilities of these tools, and basically we can become better or more productive bioinformaticians and able to have bigger breakthroughs in the future, inshallah. So before we end our sessions, I would like to thank both of our honorable speakers on today's topic, will AI replace bioinformaticians? Basically, I hope the participants, I think at the peak we have over 120 participants, so quite a good show up, and I'm sure everyone has benefited from both presentations and our Q&A session. So before we say goodbye, so let's have everyone open their webcams and everything, so that we can let the technical team take the photo for our forum today. So let's give maybe 30 seconds to 1 minute. I pass to Puan Intan to organize the photography session.
Okay, hi everyone. Kindly switch on your camera for photography session. Okay. 1, 2, 3, smile. Okay, one more. 1, 2, 3, smile. Okay, thank you. Thank you, everyone.
Please attendance link, please.
All right. So thank you.
you.
So we'll post the attendance link in the chat box. So kindly be reminded it's only available for 30 minutes. Please fill in those before the time limit. So before we end, so we have a short session with Dr. Akmal and Dr. Effirul in the breakout session. And also by INBIOSIS fellows, we invite everyone to join the session. So for those other participants, feel free to share our actually the forum is we have a recording as well on Facebook. So feel free to spread the video around and for those who cannot make it to this session. So thank you all. Take care. Have a good day.